This screencast introduces you to the cell and then discusses the plasma membrane. These topics may be found in Chapter 3 of your textbook. This screencast was designed to help you achieve the following objectives: describe the general role cells play in the human body, describe the structure and function of the plasma membrane, explain the function of cholesterol, proteins, and glycoproteins, as well as glycolipids as components of the plasma membrane. Describe the structure and function of cilia, microvilli, and flagella. Describe the structures and functions of the three types of membrane junctions. We have discussed atoms and how atoms are the building blocks of matter, including the human body. We've discussed how atoms interact with one another through chemical reactions to form molecules. Now we move to the next level of structural organization of the body, the cell. The cell is the basic unit of life. Back in the late 1600s, a curious fellow by the name of Robert Hooke looked at cork under a crude microscope. And what he saw looked something like this. The cork cells themselves had long since died, and what remained of them was the cell walls, which were basically an outline of their former glory. What Hooke saw reminded him of the living quarters of monks at a monastery, which were called cells, and it is from that observation that we have the name for the basic unit of life, the cell. The cell is responsible for carrying out all of the activities that the human body needs to sustain life. When you really think about what composes the human body, really it's cells and products produced by cells. All living things are composed of one or more cells. The main role of many cells in our bodies is to produce specific proteins. The cell contains about 60% water, so like the human body in general, the most abundant substance in a cell is water. The structural components of a cell are mainly composed of protein, therefore the most abundant elements found in a cell are carbon, hydrogen, oxygen, and nitrogen. The plasma membrane, or cell membrane, acts as a border between the cell and the extracellular environment, and it determines what enters and exits the cell. Therefore, the plasma membrane has a large impact on the metabolism of the cell. The space within the plasma membrane, excluding the nucleus, is called the cytoplasm. The cytoplasm is filled with a water-based solution called the cytosol. Specialized structures that perform distinct functions called organelles are distributed throughout the cytosol. There is great cellular diversity in the human body, over 206 types of cells as a matter of fact, and these cells differ significantly in their structure or anatomy, and that reflects differences in their main functions. However, all cells do share some common characteristics. They all have plasma membranes. They typically share very similar organelles. The organelles may be specialized and slightly different from one cell to another. All cells, at least at some point in their life cycle, have at least one nucleus. So although cells differ significantly in their structures and functions, they all share some common characteristics, features, and structures. The first major structure of the cell we are going to discuss is the plasma or cell membrane, and you can use those terms interchangeably. You're going to find that I'm very inconsistent with which term I use. The plasma or cell membrane forms the boundary of the cell. It basically separates the cytoplasm or intracellular fluid from the extracellular fluid, the fluid outside of the cell. If you look at the structure of the plasma membrane, you'll notice that it basically consists of two layers of phospholipids. It is often described as a phospholipid bilayer. Since the plasma membrane is composed mainly of two layers of phospholipids, let's briefly review the structure of a phospholipid. Recall that one end of a phospholipid is charged. It contains a phosphate and is polar. Therefore, that end of the phospholipid is hydrophilic. It is attracted to water, since water is a polar molecule, and this end of the phospholipid, which we call a head, is also polar. The other end of the phospholipid is composed of two fatty acid chains. They are nonpolar, therefore they are not attracted to water and are referred to as being hydrophobic. The phospholipids arrange themselves in two layers such that the hydrophilic heads are in contact with the water of the extracellular fluid and the water of the cytoplasm. And the hydrophobic fatty acid tails make up the interior of the plasma membrane. This arrangement of phospholipids is one of the reasons why the plasma membrane is selectively permeable, meaning it allows some substances to cross it easily and others it excludes. The hydrophobic interior of the cell will not allow polar or charged particles to cross. So ions, amino acids, mono and disaccharides cannot easily cross the plasma membrane. Nonpolar molecules, molecules very similar to the fatty acid tails that make up the interior of the plasma membrane, can easily pass through the plasma membrane. Phospholipids are not the only components of plasma membranes. Other molecules play very important roles as components as well, and one of those molecules is cholesterol. Cholesterol is embedded in the phospholipid bilayer, and it helps maintain the structure and fluidity of the plasma membrane as temperatures fluctuate. Body temperature will move up and down over time due to changes in the activity of the body as well as environmental temperature. And as you are well aware, lipids are responsive to temperature changes in terms of their physical characteristics. Think about what happens to a stick of butter if you remove it from the refrigerator and allow it to sit out on the kitchen counter for an hour or so. The cholesterol molecule helps stabilize the plasma membrane as temperatures fluctuate in a similar manner that rebar, the mesh of wires found in concrete, help stabilize concrete as temperatures fluctuate. Proteins play very important roles in the plasma membranes. The proteins are shown here in purple. There are two types of proteins associated with the plasma membrane. Some are only found at the surface of the plasma membrane, and they're called peripheral proteins, while others pass completely through both phospholipid bilayers, and those are called integral proteins. Some proteins act as enzymes, catalyzing reactions at the surface of the plasma membrane. Others move substances across the plasma membrane transporting substances either out of the cell or into the cell. They are known as carriers and channels. And still, other proteins act as anchor points for the cytoskeleton. Some proteins of the plasma membrane have a carbohydrate component attached to them and are referred to as glycoproteins. Glycoproteins are used as receptors, binding sites for immune cells, bacteria, viruses, and some toxins. Some phospholipids of the plasma membrane also have a carbohydrate component attached to them and are referred to as glycolipids. The carbohydrate components of glycolipids and glycoproteins extend from the extracellular surface of the plasma membrane and form a sugary coat. This sugary coat has several important functions. One is to protect the cell from mechanical damage. The sugary coat sort of acts like a bumper. And two, since the specific pattern of the carbohydrates of the sugary coat is determined by the cell's DNA, your immune system uses the sugary coat to distinguish the cells of your body from foreign cells. So you could think of the sugary coat as a big "Hello, my name is" sticker. The plasma membrane of some cells may be formed into specialized projections, including microvilli, cilia, and flagella. Let's talk first about microvilli. To find cells with microvilli, we have to search in a location where a lot of absorption occurs, because the function of microvilli is to increase surface area to facilitate absorption. One of those places is the small intestine. The small intestine is where most nutrients are absorbed from the GI tract into the blood. Now, the small intestine is thrown into these finger-like folds called villi. You're looking at a single villus here. Now, this is a villus, not a microvillus, but a villus. You can see a villus under relatively low power under a microscope. To see the microvilli, since the microvilli are extensions of the plasma membrane, we have to look at the small intestine under high magnification. So if we were to focus on a portion of the villus here and increase magnification such that we could see the individual cells, then you can see the microvilli here. Microvilli are also called a brush border because they sort of look like the bristles of a brush. Cilia are much larger structures compared to microvilli. You can view them under medium power under the microscope. The function of cilia are to move substances across the surface of the cell. One location where you can find cilia is in the lining of the trachea. Mucus covering the lining of the trachea trap dust particles, bacteria, and other little nasties that come in on the air that we breathe, and they're trapped in the mucus, and the cilia beat to move that mucus with all those trapped particles away from the lungs back toward the pharynx or throat. A flagellum is a large tail-like membrane extension. It's in the human body, it's only found on the male sperm cell. It beats back and forth like a whip, and it propels the sperm cell forward. Plasma membranes of adjacent cells often form intracellular junctions with one another. Intracellular junctions is discussed in your book under epithelial tissues. However, as they are specializations of the plasma membrane, I choose to discuss them here. There are three intracellular junctions that you need to be familiar with. They are tight junctions, desmosomes, and gap junctions. Tight junctions are impermeable junctions. They fuse the plasma membranes of adjacent cells together and prevent any substances from moving down between the adjacent cells. In the small intestine, for example, these junctions prevent digestive enzymes from seeping into the bloodstream. Desmosomes are anchoring junctions. These junctions help hold adjacent cells together and resist being pulled apart. You find desmosomes in parts of the body that receive a lot of abrasion, such as the cells of the epidermis, or the outer layer of skin. Notice the intermediate filaments that act as guy wires holding the adjacent cells together. Gap junctions are communicating junctions. They contain little pores called connexons, which allow small molecules and ions to move from one cell to the adjacent cell. This functions to allow communication from one cell to the adjacent cell. Now let's review the objectives the screencast was designed to help you achieve. Describe the general role cells play in the human body. Describe the structure and function of the plasma membrane. Explain the function of cholesterol, proteins, and glycoproteins and glycolipids as components of the plasma membrane. Describe the structure and function of cilia, microvilli, and flagella. Describe the structures and functions of the three types of membrane junctions. The topic of the next screencast will be the cytoplasm and nucleus.